Hello again, everybody. Mr. Rumlar here. Today I want to talk to you a little bit about traits and how these traits are inherited from one generation to the next. Before we begin, I want to make sure that we have a clear definition of what we mean by the word trait. A trait is any sort of specific characteristic that varies amongst individuals. So this could be things like, hair color, eye color, how tall you are, whether or not you have certain genetic disorders, anything that makes you different from anybody else is a trait. And these traits are carried in our DNA, which is something that we all know, we all take for granted, but this was not always a well-known idea. And so what we're going to talk about here in the next couple videos is how that DNA leads to the formation of these traits. And again, before we get into that, it's important to lay a little bit of foundation. In our last unit, we talked about cells and the difference between prokaryotic and eukaryotic cells and how there are specialized cells to do specialized jobs. But I want to focus on the two main divisions of cells in living things, which is gametes versus somatic cells. So somatic cells are the cells that you're already familiar with. These are the things that make up most of our bodies. The two really important characteristics about these are that, number one, they're diploid, which means they have two copies of the DNA. You get one copy from your mom, and you get one copy from your dad. The other important thing is that these cells are produced in the process of mitosis, which I'm sure you've all heard of before. So these are things like nerve cells, muscle cells, epithelial cells that make up our skin and the linings of our digestive system, things like that. The other main type of cells are gametes. And the two really important things about gametes is that first, they're haploid, which means they have one copy of the DNA. And this one copy is kind of a shuffled up version of DNA from your mom and your dad. The other thing about gametes is that they're produced in a process called meiosis, which is similar to mitosis but has some very important distinctions. And we'll talk a lot more about that process and the significance of haploid versus diploid as we move on. But of course, in animals, what we're talking about here with gametes are sperm cells and egg cells. With plants, it's a little bit different, but all of these topics still apply. So in the 1800s, mid-1800s is when the science of genetics was being studied and when inheritance was being studied by a man named Gregor Mendel. Moving into the 1900s, the idea of genetics was pretty well understood, but how that worked, how traits were inherited, was not very well understood. And there were two dominant thoughts in the scientific community. Some scientists thought that proteins were molecules that were transferring this inheritance, and other scientists believed that it was this new molecule that had been discovered. They didn't even have a name for it yet, but it's what we now know as DNA. And there were three really crucial pieces of evidence to point towards DNA being this molecule of inheritance. Number one, it was always found in the right place. All cells that they looked at had DNA, and it was generally found in the same location. It was always found in the right amounts. Different species have different amounts of DNA. And then it varied in the content from species to species. So these were three really important distinctions that pointed towards DNA being the molecule of inheritance. But like any good scientists, they didn't just look at that information and form a conclusion. They conducted some experiments. So I'm going to talk with you about three really key important experiments involved in the discovery of DNA. I don't expect you to memorize the dates and the names, but what I do want you to get from these is if you read about these studies again, you should be able to understand what the main conclusions were. So in 1928, a scientist by the name of Frederick Griffith was studying the bacteria that causes pneumonia. And what he found was there were two strains of pneumonia bacteria. There was one that he called the R, or the rough strain, and the other one was the S, or the smooth strain. And the rough and the smooth just refers to how these bacteria grew in colonies when he grew them in the lab. The really important part is the rough version of pneumonia causes the disease. The smooth version does not cause the disease. So what that means is, in the laboratory, if they injected the rough strain into rats, they got pneumonia, and unfortunately, they did not survive. If the smooth strain was injected into rats, they were perfectly healthy and had no problems. So the next step in this experiment, he took some of these bacteria that cause infection and he boiled them. He heated them up over a flame and effectively killed the bacteria. And by doing so, when he injected those into the rats, they survived. And then the really important part of this experiment, he took these bacteria that had been killed by boiling them and he mixed them with these smooth bacteria, the ones that don't cause illness. And the result from that was these bacteria went through a process we call transformation. And these previously harmless bacteria were now able to cause infection, and when they were injected into the rats did not survive. And so the main conclusion here was that there was some chemical substance from one cell that can cause transformation in another. All this was showing was that the mechanism of inheritance does in fact work. Years later, in 1944, a scientist by the name of Oswald Avery and his team kind of took this experiment the next step. So they took those bacteria that were killed by boiling, and then they treated them with a series of different enzymes. They treated them with enzymes that dissolved RNA, which is a type of nucleic acid. They used enzymes that broke down proteins, and they used enzymes that broke down DNA. And what they found was that when they dissolved the RNA in this solution, you still got transformation of the bacteria, the rats still got the disease, and they still died. If they used enzymes that break down protein, same thing. The transformation still occurred, the rats got the disease, and they did not survive. However, when they used an enzyme that breaks down DNA, the transformation did not occur. They injected this solution into the rats, and the rats survived. So, what did they learn? They learned that they could eliminate these possibilities of RNA and protein because when they removed those, the transformation still occurred. They gained positive evidence that it was DNA that was causing that transformation. So ultimately, they came to the conclusion that the transforming substance is DNA. And now I know you're thinking to yourself, this all makes sense, but you have to remember that this knowledge that we have now was not always available to everybody. So one last experiment to kind of solidify all these results was conducted in 1952 by the scientists Hershey and Chase. And they used a specific type of virus called a bacteriophage. So viruses are very simple in that they just have kind of this coating that's made out of protein, and then inside they have DNA. And so they grew these viruses in solutions, and the first group was grown where they used radioactive substances to label the DNA. And then in the second group, they used radioactive substances to label the protein. And long story short, they grew these viruses, they used them to infect bacteria, and they found that the DNA ended up in the bacteria that got infected, whereas the protein did not get incorporated into those infected bacteria. So the ultimate conclusion here was that DNA was in fact the molecule causing this sort of change. Now, there were two other key pieces of information in the discovery of DNA. The first came from a scientist named Rosalind Franklin, and she worked in a field called X-ray diffraction, where they would take crystallized molecules and shine X-rays through them and then look at the patterns to determine features of their shape. So this picture is the actual X-ray diffraction photo of DNA that she created. And this X pattern here in the middle and these bands of lines gave her very important clues as to the shape of DNA, which we'll get to here in just a second. The other major discovery was done by a scientist named Erwin Chargaff, and he discovered that in every sample of DNA that he looked at, the amount of adenine was always equal to thymine, and the amount of guanine was always equal to the amount of cytosine. And so he came up with what we now refer to as our base pairing rules, which say that A always pairs up with T, and C always pairs up with G. And so Watson and Crick, who are the most famous scientists in this story, they kind of took all of those pieces of the puzzle and put them together. They used Chargaff's rule to establish what we now refer to as our base pairing rules, and they used the information from Rosalind Franklin to identify DNA as a double-stranded helix. So what do we mean by that? The double-stranded part comes from the fact that there's 1, 2 strands that make up DNA, and the helix is just kind of that spiral staircase shape. Specifically, DNA is a right-handed helix, but that's not really important for you guys to know. The other significant thing about DNA, and when we talk about how DNA copies itself, this will become really important, is that these bonds that hold the side together are really, really strong. But these bonds in the middle that kind of form what look almost like rungs of a ladder, these are what we call hydrogen bonds. And they're really, really weak. They're easy to break. And we'll talk again when we get to DNA replication about why that's important. Okay, so another distinction that we need to make here is how these three terms relate: DNA, genes, and chromosomes. And so I like to use this as an analogy. Years ago, I read all 7 of the Harry Potter books. And DNA, our genetic information, is kind of similar to a series of books in that each of this whole story is broken up into 7 distinct volumes, and then each volume is divided into individual chapters. Our DNA is very similar. Our DNA is broken up into 43 volumes that we call chromosomes. Those chromosomes would then be broken up into chapters that we call genes, and those genes are made up of words that are the DNA. And so that's kind of what this image is showing you here. We've got DNA, which is this whole long line. DNA can be divided into segments, and each of those segments is known as a gene. And then all of those segments together make up a chromosome. One common misconception, people think that the DNA in our cells is one big, long, continuous molecule, but in fact, it's actually 46 individual separate molecules in every single nucleus of our body. So genes are what code for proteins in our DNA. It's something that we refer to as the central dogma that says that DNA codes for something called RNA, which then leads to the production of proteins, right? And so that's broken down into 2 main steps. And we're going to keep this really simple. The first step of making a protein out of DNA is transcription. And transcription is simply when we take our DNA and we make a specific copy of the one gene that we want to turn into a protein. And so that copy is something that we call mRNA. The second step of this process is called translation. And translation is where that piece of mRNA is actually decoded into a physical protein. So the 2 steps, we make a copy of the piece of DNA that we want, and then we take that copy and use it to make a protein. That's pretty much it. We'll go into much more detail in my AP biology class, but for this one, we're just going to keep it very, very simple. The important thing here is, how do we change these proteins? That's how we get different traits, is by having different versions of DNA. And there's 2 main ways that can happen. The first is we can create changes in the actual DNA. This can happen through mutation. So any sort of change in DNA that's caused by mistakes in replication, ultraviolet radiation, chemicals, things like that. Anything that changes your DNA is a mutation. DNA can also be changed through sexual reproduction. And again, this is where that idea or that process of meiosis is going to come into play, which we'll talk about in more detail later on. There's also a relatively new area of study out now called epigenetics. And this is a study of how DNA can change within our lifetime. And there's a couple different ways that can happen. Again, you can have mutations that change our DNA during our lifetime. But there's also this idea that your genes can be expressed in different ways. So when we take DNA and use it to make proteins, we call that gene expression. And there used to be this idea that was called the single gene, single protein idea. And it was thought that one gene had the instructions for making a specific protein. The field of epigenetics is now telling us that one gene can actually make multiple different proteins depending on the chemical environment of the cell that it's made and some other more detailed factors. But the take home here is that DNA can be changed in order to make different types of proteins. And so since DNA contains the code for making proteins, any change in that strand of DNA could ultimately lead to a change in the protein that's being made. It's like taking a recipe for cupcakes or for a cake and going in there and changing the part that says sugar and replacing it with the word salt. Obviously, you're going to get a very different tasting cookie or a very different tasting cake by changing the instructions. Same thing with DNA. If you change the instructions, you're going to change the product. And the last thing I want to talk about today is biotechnology. Like, what can we use all of this information for? And there's kind of a lot of different topics, but ones that really hit home with me are, number one, the Human Genome Project, which we're going to talk about more in class. But this was a project done years ago where scientists collectively got together and mapped out the human genome. And what that means is they identified key genes and where those genes lie in the DNA, which allows us to better study those genes and things like diseases and other things that can be impacted by those genes. Our knowledge of DNA and traits can also be used for cloning. We can identify specific genes, we can clone those genes, we can study their action in different organisms. There's a lot of stuff we can do with that. We can use biotechnology and our knowledge of DNA and traits to work with stem cells, which is another topic we're going to go into great depth in class. But stem cells are cells that are unspecialized, and they can be given instructions to become any type of cell. GMOs, you see this a lot in the news right now, but our understanding of DNA and traits can be used to influence the technology of genetic modified organisms. We can identify traits that we find beneficial and transfer those traits from one organism to another. So that's it for today about traits. Our next video, we're going to get into how these traits can be varied, but I hope you found this information useful.